Hi, good afternoon, everyone, and welcome to another episode of the Botany Bistro. We're so glad that you're here. Your table is waiting for you. We have a lot to talk about today. If this is your first time on the Bistro, thank you so much for coming. My name is Mary Dudley, and I'm the Ecology Education Manager here at the Civic Garden Center. We're located in Avondale, which is in the Cincinnati region of Ohio, and it's just so beautiful. Spring has sprung, and as you can see, the lovely redbud behind me is in full bloom. If you have a redbud in your yard, be sure to try some of the flowers. They're quite delicious. I am a trained botanist, and I wanted to start Botany Bistro because my botanical knowledge has really set me up for success in my career and in my personal life. The knowledge about being able to think about the processes of plants, to identify issues that might be happening and troubleshoot those issues with solutions that will actually be effective, not only for your wallet, but also in the long term sustainably, is very important as we move forward as a society. And so I'm giving this information to you. I hope that you enjoy it. I'm always happy to answer questions. So if you want to contact me, you can do that through the Civic Garden Center. And you can also post things to our social media and ask questions, and I'd be happy to continue the conversation. This is our 23rd episode of the Bistro. What have we been up to on the Bistro? It is episode 23 after all. Where have we come from? Well, we have talked about morphology, anatomy, taxonomy, systematics, physiology, pathology, genetics and breeding, paleobotany, ethnobotany, and agronomy. We're going to be reviewing some of the things that came up in some of those earlier episodes in our chat today, because really the field of botany is quite wide, but it does have overlap. And so as we go through this journey together, it's been neat to see how things all connect. If you want to catch up, be sure to visit those archive pages. Otherwise, we're going to jump in and get into biotechnology. As far as plants are concerned, I'm not going to be covering biotechnology in other disciplines today, but it is something that's used in several other fields as well. It's lunchtime. What's everybody eating? Today, I have a delicious eggplant and soba noodle dish that my husband made. Thank you so much, honey. And I am just smelling all those wonderful aromas. It has some sesame seeds on it and all of the just eggplant goodness that soaks up all the juice that it was sauteed in. So I'm very excited to have this. Our lunch always has something to do with our topic, and so today I will mention one of these ingredients again, so make sure you keep your ears open so that you can find out how my lunch connects to our talk today. Our lesson's going to discuss the introduction of biotechnology, and then we're going to talk about GMOs. We're also going to be talking about other forms of biotechnology, including tissue culture and cryopreservation. We're leading into part 2, which will be on April 19th. I'll have a special guest with me that day, Dr. Philpott. She utilizes lots of different biotechnology methods in her work. So that's going to be really exciting. So make sure you don't miss that in a couple of weeks. We always have a little bit of homework, and so your homework for this session is to tell us about a GMO that you might create on your own, or just imaginary, or maybe something that you already are impressed by that's come out of the scientific community. And so think about that, share it with us on social media at Civic Garden. All right, so I think it's time to get into the meat, biotechnology. Now, this might be a concept that you know a bit about, maybe not that much, but I do think that this is a topic that has a lot of people scratching their heads or possibly induces some anxiety. So I just want to let you know what we're going to be focused on today and how we're going to approach this. It's very important for us to have all the information available to us before we make decisions. And biotechnology is no different. So when we are considering how biotechnology fits into our lives, it's important for us to think about what it actually means. And so putting aside maybe personal opinions about what we think it means or what it includes, really our working definition today is that it is a range of tools that alter living organisms. So we are talking about several different methods that are employed to alter the genetic composition of a particular organism. And so I would equate this kind of to how I feel about driving my car. It's very functional. I use it every day. I don't necessarily know how to fix it. I think I have a broad concept on how my car operates. But it's a tool that I appreciate using. And just because I'm not 100% sure about all of the details about how it works does not mean that I am afraid of it. I want to know information so that I can make an informed decision as a consumer. And so biotechnology, in my opinion, is very similar to that. I might not know all the specific details, but at the end of the day, biotechnology is a huge part of our food system here in the United States and around the world. And having the information that we need to be informed is really important. Do you think that you interact with GMOs on a daily basis? And what does that mean? Genetically modified organism is a GMO. And so there are some people that will say, oh, well, that goes back to the dawn of agriculture, when we first started selecting for plants that could grow in our region. But really, when we talk about GMOs in the case of biotechnology, we're talking about more recent technological advancements. And so I'll go through a little bit of a history. All of this kind of hinges on the understanding of DNA. And so we focused on that a little bit more in plant breeding and genetics. I will defer to that session a few times that you can go back and rewatch, and so that you can get a little bit more caught up. DNA is found in all living cells. And so you have it, I have it, plants have it. And understanding how that cell structure works helps us understand how we can manipulate and change it and study it and understand a little bit more about the genetic makeup of that individual. And so DNA in your food, you bet. It's a long molecule that contains genetic instructions that is used in the development and functioning of all known living organisms. It can be seen with the naked eye when collected from thousands of cells. I'll give you a little tutorial on how to do this at home if you want to give it a try. It's taken a long time for us to be able to have the equipment to be able to see this, to be able to manipulate it, to be able to measure it. But now that we have that equipment, really that's not slowing down. And so we are in a really amazing time of advancement right now. Sometimes I feel like it goes a little too fast for my taste. And I'm like, oh, should we pump the brakes? But no one's going to stop doing these studies anytime soon. But how can you see DNA at home? So I, don't have a very fancy microscope at home. Maybe some of you do. But you can actually do an experiment where you can extract DNA. And I've done this with high school students before, and it's been a lot of fun. And so you can use strawberries. And we like to use strawberries because their cells break open fairly easily. And they're octoploid, so they have 8 copies of each chromosome, which leads to a lot of DNA. So if you're extracting DNA and you're trying to look at it without a microscope, this is a good way to get quite a bit of DNA. What you're going to do is you mash the strawberries to break the cell walls open and expose those inner membranes. And then you're going to mix that with an extracting solution, which has soap, salt, and water in it. The soap in the solution dissolves the lipid bilayers, which are those inner membranes of the cells and nuclei, which expose the DNA. The salt breaks up the proteins bound to the DNA and helps the DNA solidify. Filtering the mixture can kind of get rid of the strawberry parts that are bigger than DNA, and adding this cold rubbing alcohol precipitates the DNA, which means that it kind of comes out of that mash up solution, makes it visible. And so what you really see at the end of this is like something that looks kind of like boogers, like white kind of bubbly boogers. And that's DNA. I mean, you just mashed up the cell walls, you broke those membranes down, and then you were able to precipitate DNA out of a plant. So give that a try. I definitely recommend using a nice high quality rubbing alcohol to do this. If you are using detergent, I would use one that is clear so that it doesn't interfere with the colors that you're seeing. Now, it won't kind of stay in that state for a long time, so it's kind of something that you look at and then maybe you're tossing it. But if you are interested, there's more research you could look up strawberry DNA extraction is what you would search for. But yes, there's DNA in our food. There's two different methods I'm going to discuss just briefly, commonly used in biotechnology studies. And one is called gel electrophoresis. It's a laboratory method that's used to separate mixtures of DNA, RNA, or proteins according to molecular size. So during gel electrophoresis, you have a gel or an agar. Usually it's a little kind of square rectangle. And in one side of it, you have wells, which is where you can pipette or you can add some material. And then you put that into a liquid that has a current flowing through it. That current is running usually pretty low. You can set it different voltages. And so you're, kind of slowly pushing these molecules forward. And so the pores that are in the agar solution that you're creating, the medium that you're using, will also determine how quickly this process goes. And so we have different opportunities to create different solutions, mediums that we're using, so that we can really sort through what's going on, depending on what species we're looking at. And so the molecules traveling through the pores in the gel at a speed that is inversely related to their lengths, which means that a small DNA molecule will travel a greater distance through the gel than a large DNA molecule. So just to put this in perspective, you've probably seen like a black and white fuzzy image of bars running up and down in like a vertical column. And maybe it's a darker kind of bar and then like a little line. These are where the different molecules will sort out. And so if you're thinking about forensics, people will use DNA analysis in forensics. And so they'll have a match that they're matching, and then they'll run their gel, and then they'll put those two together. Okay, are they similar? Are they not similar? Am I seeing similar markers? Do I have any kind of correlation between these two? And so that is gel electrophoresis. These are things that you can buy fairly inexpensively from Carolina Biological Supply or another at-home science provider. But running your own DNA gels is totally possible, and pipetting is a lot of fun. The trick with gel electrophoresis is that you just need to make sure that you remember that the gel is gel, kind of like Jell-O consistency, and so your pipette is very sharp on the tip. And so if you poke through the well and you get to the and you poke through the gel, then it kind of ruins the whole thing. So you do want to be careful and just practice. But if it doesn't go well your first time, it's okay. It happens to all of us. Another term that you might hear is PCR, and that abbreviation stands for polymerase chain reaction. It's a laboratory technique for rapidly producing or amplifying copies of a specific segment of DNA. When scientists are thinking about altering part of a genome, and they're thinking about how they can introduce new genes to create new transgenic organisms, they probably want a lot of opportunities to try all that, right? Like, so they appreciate having many opportunities to give different solutions and different combinations a go. And so PCR involves using short synthetic DNA fragments called primers to select a segment of the genome to be amplified, and then multiple rounds of this DNA synthesis occurs to amplify that segment until they've reached the amount that they would like to use. Biotechnology can provide us with tools that make production of our food a little bit less expensive on some ends and more manageable. And how can that be? Let's think about how that happens. Some crops can be engineered to tolerate specific herbicides, which can make weed control simpler and more efficient. And so an example of that is the Roundup Ready soybean. So in 1996, Monsanto created this genetically engineered crop which is resistant to the very common weed killer, broad spectrum, using it in a lot of different applications. This is what we're applying now to try to reduce the comeback of honeysuckle after it has been removed, other invasive species. So this is a very commonly used herbicide. And so this crop is resistant to it, which means that you can spray Roundup around your fields, and it will kill everything except for this soybean. And that was really a game changer for a lot of farmers. It reduced weed competition. It made them not have to apply a cocktail of different chemicals. But at the same time, it also produced some issues where, these are living beings, these plants. And so they have pollen, they are visited by pollinators. And so you're introducing a lot of different factors into that. And so the Roundup resistance gene was being spread around and losing some of that control that you had over your crops. And so, nothing's perfect, but Roundup Ready is still being used today. There's actually Roundup Ready cotton, corn, and a couple of other crops. There is the chance of creating kind of super weeds that can be resistant to this herbicide. And so, we're appreciative of the advancements, but always looking over our shoulder, I think, to make sure that we're not having a detrimental effect. And other crops have been genetically engineered to be resistant to specific plant diseases and insect pests. When I am buying certain crops, I will look for those little letters after the plant variety name, and sometimes that will tell me if it has a certain disease resistance. There are tomato resistant varieties, there's other resistant varieties. We talked a little bit about those in our plant pathology section, but looking at those letters matters. So even if we're not quite sure what those mean, it really is in our best interest to kind of look those up. One of them that is pretty common is BT. BT stands for Bacillus thuringiensis, but you're like always going to see it say BT. And we have several crops that we grow that are BT enhanced, let's say. And so it's an insect protection, a piece of that organism that is put into these different plant cells, and it selectively targets caterpillars within the order Lepidoptera. And so there are several moth species that are harmful to corn, as well as some other crops. And also it isn't good at differentiating between the ones that we don't want and the ones that we do want. So, saving monarchs, saving some of our other caterpillar species is very important to think about when we are growing acres and acres of a genetically modified organism that includes a way to kill specifically caterpillars. But what are we trying to kill? We're trying to kill the European corn borer. This is the primary target for BT crops. And so this is a moth caterpillar that feeds on corn stalks and ears. Yield losses and cost of controlling this corn borer are estimated at over $1 billion annually. So if we're able to plant a variety that will not succumb to this borer, we are spraying less, hopefully, and we have better yield. And so we have more food security. And so there's a balancing act here. And there are people that are analyzing and surveying what's happening around these fields. And so if you're growing a milkweed patch right next to a field that's being grown in BT varieties, that might not be ideal. But the concept is that, the benefits outweigh the risks at this time. So we just have to keep our eyes open, keep our ears open, make sure that we are really paying attention to all of the effects. The plant part that contains the BT protein, which not all plant parts will have it in the same concentrations, needs to be ingested by this caterpillar. And then within minutes, the protein that it ingests binds to the gut wall and the insect stops feeding. Within a few hours, that gut wall breaks down and the bacteria in the gut invades the body cavity, and then the insect will die as bacteria multiplies in their blood. So sounds like a pretty bad way to go. And there are different species that have levels of sensitivity to the BT protein as well. So hopefully, we are being careful with this product so that we're using it so that it has the benefits, but we're not creating, super bugs that are resistant to it. So that's just one example of something that you might find around with some letters after it that really would influence how you do other parts of your gardening. Crop production options are necessary to help countries keep pace with demands for food while reducing production cost. And so we have a couple of different organizations that regulate genetically modified organisms. The USDA and the FDA are some big ones, as well as the EPA. It's important that all these organizations are working together. Some of them regulate different parts of the process versus others, but everything that's on the market has been carefully studied and vetted. Even though it seems like it's moving pretty quickly, there are several organizations that are involved in that process. And so plants that have genes from other organisms, so like I mentioned, we're using a protein from Bacillus thuringiensis to kill an insect that's eating a plant. So that protein is from a different organism than the plant that it is inserted to, which means that it is transgenic. And so the presence of that desired gene controlling that trait can be tested at any stage of growth. It can be in seedlings when it's in the greenhouse, and we can have breeders that are able to identify whether or not that was taken up properly by the plant, and in what concentrations that it is in that plant. Producing a new crop through genetic engineering takes about 10 years on average, and $130 million. So I can extract strawberry DNA, but I'm probably not going to be creating any GMOs anytime soon. So that's where your homework comes in. We can all imagine a plant that we would create. For me, it probably would be a lettuce or other leafy green that could handle our hot summers. I love cucumbers and tomatoes in my salad, and very rarely am I able to get all three of those things to be harvested at the same time in my outdoor garden. So I think that would be something I would be interested in. There is a list of currently used approved GMO crops. We have alfalfa, apple. The Arctic apple was designed so that it would not brown as quickly after it was cut. Canola, corn, cotton, eggplant. Oh, some of you are like, oh, she's eating eggplant. So I don't know if the eggplant in my soba noodle dish today is genetically modified. Because my husband made this, he didn't save the sticker. So sorry, I'm not sure if I'm eating a GMO today. But that is on the list. Papaya, which I discussed in the plant genetics session, really creating that rainbow papaya saved the industry in Hawaii. That ring spot virus that would have overtaken that plant, would have just decimated that entire industry. And so the ability for researchers to have been able to produce a genetically modified organism that was resistant to that virus was crucial for those farmers. There is a pink flesh variety of pineapple. I have not tried that, but I would think that most people would probably understand that that is not naturally occurring. There's something going on there. And so that's a GMO on the list. There's potatoes that have different genetic modifications. There's a salmon called the Aqua Advantage. Soybeans, squash, summer squash particularly, sugar beets, and sugar cane. What does all this mean for us? Well, in 2020, which was the most recent survey data that I found, GMO soybeans made up 94% of all soybeans planted. GMO cotton made up 96% of all cotton planted, and 92% of corn planted was GMO corn. More than 95% of animals used for meat and dairy in the United States eat GMO crops. So if you are not 100% organic, then you are eating GMOs possibly on a daily basis, or wearing them, or whatever. So this is something that's in all of our lives pretty ubiquitously. We don't have a ton of labeling that is required, and so you kind of just have to look this stuff up. We've used agricultural biotechnology to protect crops from devastating diseases. Like I mentioned, that papaya ring spot virus was totally going to decimate the Hawaiian papaya industry. And so that's session 16, part 3, which aired on December 29th, if you guys want to go back and check it out. It's not a huge list, but the crops that are on that list are used for so many things. If you look up and see what they use soybeans for and the byproducts of soybeans, you will be amazed. Hundreds of products are made from that. Same with corn. And so it's very pervasive in our society to use GMOs. And so how about a little bit of history? Let's do a little timeline. And so in 1973, scientists successfully spliced a gene from one organism to move it to another, which launched the modern biotechnology era. So that's kind of the basis for what we're talking about today. I'll start it in the early 70s. And then in 1978, the first commercial product rising from the use of recombinant DNA was synthetic insulin. Before, the only way to produce insulin for human use was pig and cattle pancreatic glands. In 1982, the first biotech plant was produced, which was a tobacco plant resistant to an antibiotic. And this paved the way for beneficial traits such as insect resistance to be transferred to plants. In 1985, field trials for biotech plants that are resistant to insects, viruses, and bacteria began being held in the United States. 1986, the EPA approves the release of the first crop produced through biotechnology, which was tobacco. And this kind of launched a coordinated framework for the regulation of products derived from biotechnology and how we are trying to make sure that we're minimizing harmful risk to the rest of the environment. In 1988, the first enzyme produced from a genetically modified source was yeast to be approved for use in food, and it's used for cheese production. And prior to that, the only way to get this enzyme was from cows' stomach linings. In 1991, the USDA's Animal and Plant Health Inspection Service published guidelines for field trials of biotech crops. And in 1994, the biotech Flavor Savor tomato was developed to have more flavor and a longer shelf life than conventionally grown tomatoes. And that was approved by the FDA. So some of you might remember the Flavor Savor tomato. It had a 3-year run, and then it was taken off the market because it wasn't profitable. But most of the tomatoes that we have in our grocery stores are actually picked before they're ripe. And then they are ripened with chemical means. And so this Flavor Saver was actually able to be ripened on the vine and then picked and taken to stores, and it would be able to last longer. But just, people didn't say that it tasted better, so people weren't rushing to go get it. And there was a big uproar about it. A lot of restaurants were proud to say that they would not have that product in their menus. And so that kind of kicked off, I think, a lot of the public education that's happening, but also it went off the market. So, if people don't want it, if there's not a market for it, it won't be developed. $130 million is a lot of money to invest in something without a return. In 95, 96, biotech soybeans and maize corn were approved for sale, and biotech cotton was commercialized in the United States. Biotech crops became the most rapidly adopted technology in the history of agriculture. So this is a field that is growing very quickly, literally, and we're just breakneck speed trying to keep up with it. In 1996, farmers in 6 countries plant biotech crops on 4.2 million acres. And then in 1999, German and Swiss scientists developed golden rice, which we also talked about in a previous episode, which was fortified with beta-carotene, which stimulates production of vitamin A that could prevent some forms of blindness. So golden rice is still kind of in a gray area, back and forth, but it is starting to be grown, and hopefully will be hitting the markets, and it does have a different color. It does not look white. It is more orange, more of a yellow. So that's interesting. And then in 2000, the first entire plant genome was sequenced. Arabidopsis is a very small, kind of mustard-looking plant, but it provided research with such great insight into the genes that control specific traits. So having that fully mapped genome of this kind of nondescript plant, but was used very heavily in a lot of studies because of its fast growth, was really crucial to moving this forward. And so what are the basic steps of, kind of creating a genetically modified organism? Well, first you identify a trait that you are interested in. You have to isolate that genetic trait and then insert that trait into the genome of a different or desired organism, and then grow that engineered organism. And so that is a process that obviously takes a lot of skill, a lot of tools and techniques, and has been honed over time. Tissue culture, which is what we're going to kind of move on to next, is another opportunity for people in plant biotechnology to propagate plants. And it's a significant development for plant breeding, and also can be called micropropagation. And tissue culture permits researchers to clone plant material by using small amounts of tissues from plants of interest. And then they induce the growth of that tissue through a media. We kind of talked about gel a little bit, agar a little bit, which forms a new plant. The new plant carries the entire genetic information of the donor plant. So we're talking about a full clone. And exact copies of that plant can be produced without depending on pollinators or seeds, and can be done in a fairly short amount of time. I am excited to introduce you to Dr. Philpott next time we have a Bistro session, and she showed me her laboratory with lots of tissue culture happening, and so I'm excited to hear more about that from her. But they particularly are using this for conservation, and so endangered species that you might not want to take the whole plant home, or maybe it's not producing seeds at the time, or maybe it doesn't produce seeds. How are we going to preserve that material and keep it safe for future generations? And tissue culture is one way to do that. And so you'll hear the words in vitro, which means literally within glass. And so growing plants in little test tubes with this kind of clearish looking gel at the bottom of it, and the roots are getting right in there, and they're getting the nutrients from whatever that gel was made with. And then they're growing up in a little test tube under a light in this room. It was just really fascinating to have this indoor garden, essentially thousands of different plants, all growing in very small little tubes. And so orchids are a common tissue culture plant, if that's something that you have been interested in, is orchid culture. And it's all done under sterile conditions because fungus would love to grow on that rich nutrient media. And it can be a barrier to do tissue culture at home, but I'm going to give you a little recipe. I love doing science at home. So after you're done extracting the DNA from your strawberries, you can try a little tissue culture. And so I've got a little recipe for us here today, and it is table sugar, tap water, some all-purpose soluble fertilizer. You're going to want a stock solution of an inositol tablet, vitamin tablet with thiamine, and then some agar flakes. But you can produce your own growing medium. You just get your test tubes out, and then you would be able to cut small parts of a leaf and try to get that leaf to stimulate additional growth and roots so that you could grow a whole plant in a little jar, in a little tube. We've talked about using biotechnology to create genetically modified organisms, thinking about tissue culture for propagation, and the last section was just going to be talking about cryopreservation. Talking about conservation, we're trying to use biotechnology to support future research by conserving biodiversity. And seed banks are wonderful treasure troves of biodiversity and how we can make sure that we're trying to save that for the next generation. It's typically very cold, so you want those seeds to remain dormant. You want it to be dry. You don't want to have molds or fungus kind of getting in there. And that's a great option to preserve a lot of plant material, different seeds. But not all species can be preserved by conventional seed banking. There are seeds that have sensitivities to desiccation or being dried out, and they might not be viable after that process. And there are other seeds that would not be able to withstand the cold for extended amount of time. And so plant species that are not able to be seed banked can be successfully tissue cultured, and so you can just kind of keep that going over and over. You can also plant them obviously out in gardens and maintain outdoor preserves for them, but that can also take up a lot of space and can be hard if you are regularly hit by weather events or something like that. And so thinking about cryopreservation as an option as well. And so I think that Dr. Philpott will talk to us a little bit about cryopreservation next time. They call these plants that cannot be conserved long-term using conventional methods exceptional. Biotechnology is something that is used in the cryopreservation lab, and they are stored at ultra low temperatures. You can have lots of species in there, and they are going to be preserved, but you don't just have to preserve the seeds. You can preserve different parts of the plant, and then you can experiment on getting those out, seeing if they're viable in the tissue culture. So it all kind of works together of what those goals are. And so the Linder Center for Conservation and Research of Endangered Wildlife is what CREW stands for. And they have their cryo-biobank. And there are thousands of samples representing hundreds of animal and plant species stored in tanks of liquid nitrogen at negative 160 degrees centigrade. The plant samples include seeds, spores, shoot tips, embryos. They'd also have animal specimens. But we're talking about plants today. And their scientists are working with several other organizations to make sure that we're preserving that material. I want to leave you with just some action things that you can do to make sure that you are protecting biodiversity. We wouldn't be able to do half of these experiments if we didn't have a range of organisms to choose from. And so if we limit the biodiversity that we have on this earth, it's going to affect the ability of us to have choices. It just limits our choices. So improving biodiversity in your home landscape might include thinking about planting native plants, plants that are locally relevant to your area. Thinking about how you can encourage different pollinators, birds, butterflies, bats, bees to come and visit your space. Really supporting groups that are eager to continue the work of sustainability, and talking to neighbors and maybe asking them to plant non-GMO seed, or just asking questions, not accusatorily. A lot of us are having the same goals and we're in this together. And so I think that opening up those conversations are important. If you are trying to avoid consuming GMOs, you can be comfortable choosing something that has an organic label on it. Organic growers cannot use genetically modified organisms in their animal feed or in their practices. But understand too that we probably are interacting with GMOs in other things, especially the soybean and corn products that we're using on a day-to-day basis. And so instead of kind of demonizing this field, I think that it's time for us to get more information and then be able to make informed decisions and promote the type of science that we want to have moving forward.